So whether you're excited about the molecular level and what's happening when the protein cuts the DNA, whether you're excited about human health or agriculture, or whether you're excited about the societal implications and ethics of this technology. We're going to cover all of that today, as well as microbiology. So some of you on this call might be interested in that field. But what you see, this cartoon right here, Cas9, CRISPR-associated protein 9, that's the protein that won Jennifer the Nobel. And so by the end of this talk, you'll be an expert in this very special protein. So just to orient you, when the pandemic's done, I encourage you to come visit the IGI. We're at the intersection of Hurst and Oxford. So that's where our building is. Give you a tour of labs if you want. But just, yeah, we're right down the hill. And the IGI, we're divided into a few different program areas. So as Ben mentioned, we have a lot of projects in human health. So we're trying to cure sickle cell disease. We're interested in autoimmune diseases with UCSF. We're also, I think, fundraising to tackle some neurodegenerative diseases. We're also, another program area is advancing genome engineering. So I just showed you CRISPR-Cas9, but there's tons of other CRISPR proteins. And so we're trying to find more, engineer them, give them new functions. And then we also fund a lot of agriculture projects, both at UC Berkeley and UC Davis. So we're engineering dozens of different crops, making them disease-resistant, drought-resistant. And we're starting to focus on climate change. So how can we engineer different plants to make them more sustainable under different weather conditions? So we hope to enter that area over the next few years. Where I exist is in our public impact area. So we do education, public engagement, and policy. So we're really trying to not make public impact an afterthought, but actually embed it into the science. So if we are going to cure sickle cell, how can we engage with the sickle cell community to make sure it's affordable? What's the point in having a $2 million therapy if no one can afford it? So just to make sure we're all on the same page, every cell in your body has DNA, 6 feet of DNA, so 3 billion base pairs. These are wrapped into chromosomes, further wrapped into histones, and the same 4 letters make up all of your DNA. But sometimes one of those letters can be mutated and cause a genetic disease. So there are 1,000 different genetic diseases that are caused by mutations in a single gene. And so you can imagine with something like CRISPR, we can go in kind of like a genome surgery and fix that one gene and hopefully cure that disease. So sickle cell is an example where it's a single letter change that causes the disease. And when there's a mutation in a gene, that mutation is then carried over into the RNA, which is then carried over into the protein. So that can cause it to misfold. In the case of sickle cell disease, the single letter change results in a wrong amino acid, which kinks the protein, and it forms long fibers in our red blood cells. So that's why it can't deliver oxygen appropriately throughout the body. So a single amino acid can result in this debilitating disease. So a lot of times in my talks, I skip straight to the genome engineering aspect of CRISPR. But since this is an LBNL, Berkeley talk, we can't skip over the discovery. And so what you see on this screen is a bacteria having an awful day. So the green thing is the bacteria, and the pink purple things are bacteriophages. So these are viruses that infect bacteria. So the bacteriophage lands on the bacteria, injects its DNA or RNA, reproduces itself, and then busts out, and then lands on others and exponentially grows. So bacteria, over the course of millions of years, have evolved a range of different immune systems, an arsenal, if you will. And so that's a cool picture of a bacteriophage. Looks like a little moon landing. And this is Professor Jill Banfield. who's in a few different departments on campus, also has ties to LBNL. And she's really interested in microbial communities and finding new microbes scattered across the world in different environments. And she is one of the early CRISPR researchers who was interested in this novel immune system. So CRISPR is actually a bacterial immune system, a way that bacteria defend themselves against viruses. And it's really cool because it was the first adaptive immune system discovered in bacteria. So in your body, you have antibodies, which are adaptive immune system, right? It can keep pace with your infections. It can keep a memory of them. And with CRISPR, it's the same thing. A new phage infects a bacteria, and the CRISPR system stores a memory of that infection and uses that memory against the phage when it comes back. So I'm going to show you a quick video that nicely captures what I just mentioned, how CRISPR works, how it's a memory, how it uses itself to defend bacteria against viruses. So what you see, the brown circles are the bacteria, and now you have a bunch of different bacteriophages, viruses, infecting the bacteria. So this pink one injects its DNA into the bacteria. The blue stretch is the bacteria's own DNA. So I'm going to pause it right here. And so what just happened was the bacteria cut off a snippet of the viral DNA and put it in its own genome, like a memory. So you can, if you look in the bacteria's DNA, you can see the history of infections. You can see that this bacteria was infected by a pink virus, an orange virus, a yellow virus, a green virus, kind of like you have a vaccination card if you go to the doctor's office. So we call this the CRISPR DNA. So CRISPR stands for clustered regularly interspaced short palindromic repeats. So it's a mouthful, but that's what CRISPR stands for. So how is this then turned into a bacterial immune system? So this stretch of DNA is then transcribed into CRISPR RNA, and then other proteins come along and cut this into small segments. So now you have these segments floating around, and these CRISPR proteins then come and attach onto them. So in this case, we have Cas9, and it attaches onto that CRISPR RNA. And so now you have what we call these search complexes floating around. And if the pink phage comes back, now you have this CRISPR RNA that's ready to recognize it. So if it has a complementary base pairing, the protein then cuts the viral DNA, and other proteins come along and chew it up. So I'll show another video that captures this again, because it takes a couple times to see it, to make sense of it. But what you just saw was a CRISPR RNA that used complementary base pairing to match with DNA. And that's how it's able to search through thousands, millions of different base pairs of DNA to find a very specific match. And so it was studying this bacterial immune system that Jennifer and her collaborator, Emmanuel Charpentier in Europe, realized that you could program the CRISPR-Cas9 protein to target any sequence of DNA that you wanted. All you had to do was swap out the guide RNA. So it was the guide RNA that's finding the DNA. So if you just swap out that guide RNA sequence, then you can program the Cas9 protein to target any sequence of DNA. So it's really simple. Now, CRISPR isn't the first genome editing technology. There's something called TALENs, zinc fingers, meganucleases. These have been around for a while, but it's not, in those cases, it's not RNA that's recognizing the DNA, it's the actual protein itself. So if you wanted to target a new sequence of DNA, you actually had to reprogram the protein each time, which takes a lot of time, decent amount of money, so just a lot of resources to do. But swapping out RNA is really cheap. And it's so easy that high schoolers now are doing it. We teach a CRISPR course at Berkeley. So we do a summer course where undergrads can do this. So now, okay, just forget what you just saw in a bacteria cell. Now we're taking that CRISPR protein and we're putting it into a human cell. So now we're a scientist and we want to edit human DNA. What are we going to do? So what you see in front of you is a human cell. It's a lot bigger than a bacteria cell. And like I had mentioned before, there's 3 billion base pairs. So we're going to zoom into the nucleus, and it's super crowded. You have your DNA wrapped around histones, really compact. So now you have the Cas9 protein floating around. And let's say you're a scientist and you're really interested in locating that yellow sequence of DNA and cutting it. So you design your guide RNA so that it matches that sequence of DNA. The protein unwinds the DNA and makes a cut. And so now it's up to the scientist what to do next. Do you want to insert a sequence of DNA? You want to remove DNA? And in this video, they're inserting a sequence of DNA. Now, one important thing to note, some people say CRISPR is cut and paste. Really, the CRISPR-Cas9 protein is just cutting. It's not involved in the pasting process. It's just the molecular scissors. And what you just saw there were these repair enzymes that came along and pasted in a new sequence of DNA. It's really important to note that when you go outside into the UV rays, your DNA is getting cut up all the time, shredded. And so your body has evolved many sophisticated repair pathways and repair proteins to fix those breaks. So when the CRISPR protein comes along and makes a cut, it's your body's natural repair proteins that then go and fix it. So a lot of the research right now, a lot of the labs across the world, a lot of funders are focused on this repair process, because it's by understanding the repair process and biasing it where we're able to insert DNA or remove DNA or do what we want. So now you're the scientist, and how are you going to do a CRISPR experiment? Well, the first thing to do is identify where in the genome you want to cut. So that red base pair right there is actually the sickle cell mutation, the real sickle cell letter change. And so you find 20 letters of DNA, which I've identified in bold. So the actual sequence of guide RNA that sticks out of the protein is 20 letters. So you have 20 letters that you're able to work with. And so you're going to design that guide RNA. And now there's companies that make this for you pretty cheap. The most well-known is Synthigo, there's IDT, there's all these companies that will make these guide RNAs for you. So you just go on your computer and you type in, I want a guide RNA that's AUCCCUG, blah, 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 blah. And so we called this 20-letter stretch the programmable sequence, because those are the only letters that bind to the DNA. The other stem loops that you see at the top, we don't touch that. That's the part of the RNA that the protein holds on to like a handle. And then in a test tube, you can mix that with your Cas9 protein and it forms a complex. And then you can add that into the cell and it finds the matching complementary sequence of DNA, unwinds it, and it always makes a cut at the same spot, 3 letters in. And then the Cas9 protein will get knocked off. So that's a snapshot. If you were a CRISPR scientist and you wanted to edit some DNA, that's roughly how you would go about doing it. And I made it sound really easy. Can't really do this in your garage. You do need equipment. But if you have the right equipment, if you're in a lab, especially at Berkeley, you can easily do that experiment. If you want to learn more, I would encourage you to check out the documentary Human Nature. It's on Netflix now, I think. And it highlights 3 Berkeley faculty that have been pivotal in the CRISPR discovery process. So it's just a fantastic documentary. This is a really cool video, one of my favorite, where you actually get to see Cas9 in action. So this is the Cas9 protein. This is the DNA. And you're actually going to see it cut it. So there it goes. Cut the DNA. So that's pretty cool. That's atomic force microscopy. If you people on this call know what that is. So why did CRISPR win the Nobel? As far as I know, you can't go to the doctor's office right now and get a CRISPR treatment. You aren't eating CRISPR food in the supermarket yet, right? CRISPR hasn't reached the average person. Why did it win the Nobel? Now, I would make the argument that it's because it's revolutionized science. It's revolutionized basic research. What do I mean by that? So we all know these organisms on this slide. We have frogs, zebrafish, mice, flies, C. elegans, or Arabidopsis. These are all model organisms, and we've been working with these for 100 years, right? How many times have we cured cancer in a mouse? And this is because we've kind of been constrained to these organisms. It's really difficult to edit the DNA of other organisms. But now with CRISPR, all these organisms I'm showing right now have been edited. Dogs, chickens, ferrets, tons of fish, tons of butterflies, monkeys, all these microbes and crops. We were keeping track at the Institute for a while, but it's hundreds now. I don't even know, 400 or 500 different species. And there's very little barrier to entry. You can pretty much edit anything. And so now we can answer questions that we weren't able to answer before. We can ask questions that we didn't think we could ask before. One really cool application of CRISPR is in this top left corner with this squid. It's been, squids have really fascinating nervous systems. And we haven't been able to really study them because you can't start adding electrodes into squids. Just the physiology doesn't work like that. It's difficult to work with. So people used CRISPR to edit genes to figure out, better understand the nervous system of the squid. There was a researcher at Berkeley who was really interested in editing butterflies. So several labs have edited the DNA of butterflies to repaint butterfly wings, understand pattern formation, the evolution of butterflies. On the top right, that was the first reptile edited with CRISPR. And another really cool example on the bottom is an insect that was edited with CRISPR to understand the social interactions, right? So insects communicate in very fascinating ways. And so they were editing genes to understand the connection between biology and how those insects were communicating with one another. So by editing the genes, could you impact the social complex of their colony? Now, at the IGI, we do a ton of agriculture research. There's, editing crops is there's there can be opposition to it. So I'll, lay out why are scientists at the IGI around the world trying to edit the DNA of crops. So the first example I'll give is cacao. So we have a team at IGI who's really interested in editing the DNA of cacao, primarily for disease resistance. So as climates change, the farmers are constrained to where they can grow cacao. Like maybe you used to be able to grow it in across thousands of acres, but as climate changes, maybe now you're constrained to a certain elevation or a certain plot of land. And what that means is that now if a disease comes, say it's a fungus or an insect, it, instead of wiping out a little bit of the cacao, it might wipe out all of it, because now it's the plants are closer together in these farms. So these scientists were interested in, I think, a fungus and a virus. So they're working actually with Mars because Mars has, they don't want to see all the cacao get destroyed, to try to figure out how can they edit the cacao to withstand these viruses and fungus. And the second example, I've been interacting a lot with these scientists and learning a lot about the motivation behind their research. So this is cassava. It goes by a bunch of different names. is a primary component of tapioca, but it's a staple crop in a lot of African countries. But it naturally makes cyanide. And if it's not cooked properly, the cyanide can build up in your body and damage your nervous system. And so people who have cyanide poisoning have difficulty walking or can't walk at all. And you can imagine in countries maybe where there's civil unrest or droughts and there's issues in accessing water, they're not able to boil the cassava correctly or prepare it correctly, and thus leading to cyanide poisoning. And so they're editing the cassava to manipulate the cyanide pathway. And maybe not turn it off completely, but can we modulate it? Can we just have it produce less cyanide? What's really important to study in addition to the science is why does it make cyanide to begin with? Maybe it's, so insects don't attack the cassava plants. So in addition to the gene editing, they're working with other researchers who are studying cassava and its natural environment. So I think those are, the cacao and cassava are really cool examples. I think also because where they're grown. So we're working on projects that don't really impact the US. We're working on projects that impact African countries or Southeast Asian countries. One of our big climate change projects is going to focus on rice, for example. But what that also means is we need to interact with those countries and the farmers over there, understand, do they even want this edited? How would they use it? What's the intellectual property? Like, will they be able to access the seeds? Who owns the seeds? So those types of questions. So now let's talk about medicine. This is definitely an area of interest for a lot of people. We all know someone who might, has a genetic disease. Maybe some of us have genetic diseases. So what is the status of genome editing in medicine? What are our expectations? The time scale of this technology. I've brought up sickle cell a few times because it has become the poster child of gene editing. Some people see it as not low-hanging fruit, but a great first step. And I'll explain why. So we've known for over 100 years. We've known that it's a genetic disease. People for a long time have known the exact letter that's mutated to cause sickle cell disease, but we've never had the tool to go in and correct that mutation. We also know that inside your bone marrow are the stem cells that produce your red blood cells. So if we can find a way to get to those bone marrow cells and we correct the mutation, then in theory, you should be able to produce round red blood cells. Because what happens in sickle cell is they create these sickled shaped red blood cells which clog in your veins, are unable to transport oxygen. But there's also a lot of societal implications. The African American community has a very complicated history with the medical community. Right now, sickle cell patients, they have a lot of pain crises, and so they go to hospitals for pain medication. And sometimes doctors don't want to just keep giving patients pain medications. But there's no cure. Some people undergo bone marrow transplants, but you need a matching donor. So with CRISPR, you can imagine, well, you yourself are your donor. If we can just take your cells, edit it, and then put them back in. So there are several different groups out there working on this. The IGI is one of them. We just had our IND approved to begin clinical trials in 2021. But there are some other companies and academic groups who are also working to cure sickle cell. On this slide is Victoria Gray. So NPR has been following Victoria for, I think, 2 years now. She's really brave. I think she was the first person dosed with CRISPR in the US. And so far, they've been following her through the pandemic, and she's been feeling great. And it seems to be working, but we won't know. You want to follow these people for years to make sure that you've actually cured the disease. but it's really looking promising for sickle cell. And it's great that multiple groups are working on it because some approaches might work better than others. But what really interests me from an ethical point of view is how much is this going to cost, right? It doesn't help anyone if it costs $2 million. So is there a way we can make it affordable? And that's a very difficult question. It requires policy. It requires communication with insurance companies, all these things that are outside of the scientists' control. In addition to sickle cell, there were actually patients in the US who had CRISPR injected into their eye. So there's a genetic form of blindness, and the eye is actually a great place to inject CRISPR. You just move your eyeball over, inject a needle. Whereas with the sickle cell mutation, you had to remove your stem cells, edit them in a lab, and put them back in. So you need to be in a hospital for like a month, and it can be painful. But with this, yeah, you just inject it right into the eyeball. So bringing up both this genetic eye disease and sickle cell, these are two different delivery approaches. And so right now, a lot of funding, a lot of effort is going into how can we deliver CRISPR to the cells in the body that we need to get to. Not every genetic disease is going to be like sickle cell, where we can remove the cells from the body, edit them, and put them back in. That's not going to work for something like Huntington's disease. We can't remove the brain. So how do we get the CRISPR molecules through the blood-brain barrier? How do we get the CRISPR molecules to the heart, to the liver, to the lungs if we want cystic fibrosis to be cured? Recently, a company just injected CRISPR into the body, I think straight into the blood, to treat a liver disease. Because pretty much everything in our body gets funneled into the liver. So it's actually kind of an easier organ to get to. But this question of delivery is a difficult hurdle. Other people are working on muscular dystrophy. A lot of exciting research was done in dogs, and they're showing that you can inject the CRISPR molecules straight into the muscle. Huntington's disease, like I just mentioned, cancer. So some of you may have heard of CAR T therapy. But this approach is not going to the cells that have the cancer mutations. It's actually editing your immune cells so they can better combat the cancer. And then HIV. So people are working hard on using CRISPR to cure HIV. And it's the pandemic, so I'm going to have to bring up viral detection. Cas9 isn't the only CRISPR protein. There's Cas12, there's Cas13. At the IGI, we made a playing card deck because there's so many. There's Cas1 through, whatever, 14. There's Cas-X now, Cas-Y, Cas-Phi. They're using Greek letters. So in this example, they're using Cas12 and Cas13 to detect viruses in samples. And I'll briefly explain how they do that. One of the main companies leading this is Mammoth Biosciences, which was a spinoff from the Downer Lab. And what they're doing is you take the Cas13 protein, you give it the guide RNA that will match, say, the SARS-CoV-2 virus. And if it finds a match, it actually turns on a pair of scissors on the other side of the protein. And you can actually, in the sample, add a little fluorescent signal molecule. And this little signal molecule is a stretch of DNA or RNA, and with a fluorescent probe on one end, and it only starts glowing if it's cut. So it's a chain reaction. If the CRISPR protein finds a match, if it says, oh, there's SARS-CoV-2 in the sample, it will turn on this other pair of scissors that cuts that fluorescent signal, and it glows. So that's kind of how that works. They just teamed up with a company, and hopefully this will roll. I think they're going to start testing on Berkeley's campus soon, do a beta test. Now, the ethics of CRISPR. A lot of people want to talk about how this will impact humanity. How are we going to edit ourselves, edit our kids, generations down the road? Some of you may have heard of He Jiankui, who edited embryos in China and implanted those embryos into women, and gene-edited twins were born. What he was trying to do was he was trying to engineer HIV-resistant children, because actually there is a naturally occurring mutation in some people from Scandinavian descent that can't get HIV. They have a particular mutation. So he was trying to recreate that mutation in these two children. But when he created this YouTube video and announced it, and there was immediate backlash. One, because the science just isn't ready. It's not ready. There's still things to improve. Another thing is he wasn't transparent about it. This isn't really something you do without anyone knowing. Another thing is we have seen his patient consent forms. He definitely didn't explain to the parents what he was doing correctly. So there's a million, and the science was sloppy. He's only shared a few slides. He hasn't published a paper, but from what he shared, one of the children is a mosaic. And what that means is he edited the cells after they had already divided. And so only a fraction of the cells in their body has been edited. Also, to do this, you want to edit both copies of the chromosome. And in one of the children, it looked like he only edited one copy of the chromosome. So it's even unclear if these children are HIV resistant. Additionally, he wasn't even able to recreate the naturally occurring mutation found in these Scandinavian people. So pretty much the science was awful, and just the whole thing was awful. But it fueled a conversation. So, ethicists, regulators, scientists were, it sped up a conversation that needed to happen. And this is a conversation that we've all had been interested in, because these were all the magazines that came out when CRISPR, became a thing back in 2013, 14, 15. Everyone was thinking about blonde-haired, blue-eyed babies, and can CRISPR increase our IQ and make us run faster? And so it's I think a really important conversation to have, but also we want to keep things into perspective. There aren't going to be designer babies anytime soon for various reasons, which we can talk about later. But it's important for us to think about how we view people with different genetic traits than us. Diversity is a good thing. So where do we draw the line? Are we okay with curing Huntington's disease? Are we not okay with editing our muscles to make us stronger? It seems like right now we're not okay with editing embryos, but we're okay with editing, adults or children. So that's where I will stop for now. I would encourage you to follow us online. We have a lot of great YouTube videos, our website. You can contact me with more questions, but we can begin our Q&A.
Great. Maybe we should ask people to chat.
Yeah, and pull your slides down so we can just see each other, if that's okay.
Yep.
So you can use the chat function on the bottom. Send the chat to everyone, and we can, I'll monitor it and can ask the questions. Well, let me ask a question while we're waiting. Kevin, one of the things I didn't quite understand is how, once you've done the cut, how does it replace the section with what you want? Is it because the half that you haven't cut has the right code to put the right one in the cut, or the right gene in the cut? I probably haven't explained this right.
Can you repeat that real quick? Someone also wrote about DNA repair that I was reading. Yeah.
Why don't you go to that? That's pretty much what I wanted to say.
Yeah. Amy had an excellent point about what UV light does. I don't know if I want to read all of it. Radiation UV light versus radiation. I guess the main point I was getting at was your body has a ton of different repair mechanisms that we're still trying to unravel, and how can we use the natural repair mechanisms to our advantage when we're trying to engineer our DNA.
I think Mark Durst asked, you alluded to the repair mechanism. Doesn't it try to just fix the cut you made? And then how do you end up with a change?
Yeah. Yeah. So when we talk about CRISPR and DNA repair, we usually mention two different repair pathways. So one is called non-homologous end joining, NHEJ. And this is the cell senses a cut and it tries to quickly fix that. And in the process, it can remove some letters, add some letters, it's a little sloppy. And you know that DNA is read in 3 blocks at a time, codons, right? So if you add a letter, you've thrown off what we call the reading frame, right? You've messed that up. But that's not a bad thing. Maybe we just want to turn off a gene, and the best way to do that is to break it. Let's say you have a genetic disease that's making a protein that's toxic and it's building up in your body, and we can just turn off the gene. And so we can use this NHEJ pathway to break that gene. If we want to insert a new sequence of DNA, we use a pathway called homology-directed repair, HDR. And that's a really cool pathway because, like I was saying, you have 2 copies of each chromosome. And if one gets cut, it can use this other copy as a template. So it says, this is what it should look like. It's a homologous copy. And so instead of using the other chromosome to fix this repair, what the scientists can do is add in a template and say, don't use this chromosome. I'll supply you with the DNA that you can use so that's a way to insert DNA at that break. So when we want to insert DNA, we use homology directed repair.
Great. So Gloria has a question. How has the Cas protein and RNA gotten into the cells?
Yeah, so in the case of, say, the sickle cell example, where we're taking the stem cells out of the body, we have them in our lab, and we want to find a way to get the Cas protein into the cell. There's a couple ways you can do that. One, you can, what we call electroporate. You can give a little shock, and it opens up the pores in the cell. You can get in that way. Another way is you don't actually need to insert the protein itself. Maybe we can just insert the DNA that codes for the protein. So we're inserting the DNA, and then once the DNA makes its way to the nucleus, it makes the Cas9 and the RNA that then goes on to edit the DNA. But let's say we're interested in editing Huntington's disease. We want to get to the brain. People now are trying to use like lipid vesicles, like these little blobs of fat, to put the Cas9 in. People are using gold nanoparticles. People are using viruses. So they're engineering the virus. These AAV viruses is what they're called. And you can package DNA inside, the DNA that encodes for the CRISPR protein, and you can use that to deliver. So there's tons of different ways to try to get to the cell. And sometimes another cool way is just taking the CRISPR protein and attaching antibodies onto it to hopefully direct it to what cell type you want to go to. So there's a bunch of different tricks you can play.
Dorothy Gordon had a question. If someone is treated to cure sickle cell anemia, does the cure pass to their children?
Yeah, so I didn't mention this, but you can think of the cells in your body in 2 categories: somatic cells and germline cells. So somatic cells are your eyeball, your heart, your lungs, pretty much every cell in your body. Your germ cells are your eggs, sperm, and embryos. And so if you make an edit in germ cells, that's passed on to future generations. If you make edits in somatic cells, that's not. So if you edit, the way people are trying to cure sickle cell anemia won't be passed on to future generations if we just edit our stem cells.
Any more questions?
There's another one that came in. And Mark,
you
asking, you might have answered you alluded to repair mechanism. Doesn't it try to just fix the cut you made in situ?
Yeah, I think we
talked about that. Okay.
Yeah, but Chris and Jay have one. How did you get the edited DNA into?
Oh, okay.
I'm
Yeah.
not sure what that means.
Yeah, yeah.
It means there's so many cells in the body. How do you get it
Mhm.
into the zillions of cells that have to be fixed?
Yeah.
Uh,
So let's give an example of an instance where editing only a few of the cells works, and an instance where you want to hit as many cells as possible. So for sickle cell, you actually only need about 5 to 10% of the cells to be edited to cure the disease. That's enough to transport the oxygen, lubricate the veins. And so what that means is when we extract the stem cells, we edit them, we put them back in. You want that process to be as efficient as possible, but it doesn't have to be 100%. You don't need to get all of them. But Huntington's disease, one hurdle is you want to get to as many neurons as possible. So, people are thinking about, okay, if we inject the CRISPR using, say, a needle into the brain, how do we make it diffuse? Another example, people are interested in cystic fibrosis. When you have cystic fibrosis, your lungs are really mucousy. So how can we get the CRISPR protein and have it hit enough lung cells to cure the disease? So it's a difficult one, but it depends on the disease.
Well, let me ask a question. What?
How do you do it?
You answer the question, how do you do it? How do you get so many cells?
So one way, what people are trying to get to is, say we're using an AAV virus, or let's say we have a CRISPR protein and we've added antibodies on it to help direct it to a certain cell type. Well, we're just going to need to add enough CRISPR protein into the blood so that it can go to enough cells. So it becomes a numbers game, dosing the patient with enough of the CRISPR. If that answers your question. And for muscular dystrophy, for example, yeah, it's just a matter of, having the CRISPR protein. Your muscle cells are pretty long. And so injecting the CRISPR protein and having it diffuse to enough of the muscle cells, but you're both diffusing, height-wise and width-wise. So it's a hurdle. It's a hurdle.
Can I just ask a question? Can you hear me?
Yes. Yes.
So I had experience with someone who was having, one of these stem cell, then growing, then transplant, and they were told the young person would only have one opportunity to have this because of the kind of the chemotherapy issues of trying to knock down your body, basically, saving your eggs so you won't be sterile.
Yep.
Do you think CRISPR would be a potential future for someone who was told that, and unfortunately that case didn't work? So is CRISPR sort of the next horizon of maybe another stem cell trial or effort, and when something doesn't work?
Yeah, so I'll mention two things. So with the sickle cell example I was giving, you still run into those issues. You need to remove the stem cells from the body and edit them and put them back in. So when you give the person a drug to immobilize the stem cells, so you can take them out, and then you dose them to kill the stem cells remaining. So like your chemotherapy, edit them, put them back in. So you're still going to run into those issues you just mentioned. But what we're hoping is, this is version 1.
Right.
Version 2, what we're hoping is that you can just inject the CRISPR protein into the blood and direct it to the bone marrow stem cells. So that way you don't have to go through that chemotherapy. But one other thing I'll mention, though, is, and we've seen this in reports and studies, is that your body can since the CRISPR proteins come from bacteria, the main one people use is actually from the bacteria, Streptococcus pyogenes, which is, your strep throat bacteria, your body might have antibodies for it already and recognize it as a foreign invader. So if you do a CRISPR treatment and say it wasn't successful and you want to try something else, your body might react poorly if you try to reintroduce the CRISPR protein into your body. But what people are doing, though, is there's a bunch of different types of CRISPR proteins. So maybe you just try another CRISPR protein from another bacteria.
Thanks. Thank you.
So Amy Cronenberg had a question. What about editing? What about gene editing in germ cells resulting in chromosome loss? Is there a way around this?
That's a good question. I get asked a lot about silencing chromosomes, particularly for disorders that result from having, one too many chromosomes. I don't quite know enough about the process of chromosome loss to answer your question well.
Okay, Howard had a question. Howard Harayama had a question. How long does it take for changes to manifest some of the examples cited?
Great question. And that varies. I think with the sickle cell example, they noticed results in, gosh, what was it? I think in under 40 days. So when they filed for the clinical trial, for the stage 1 clinical trial, they needed to see some positive results within 40 days, or else the trial would be off. But that's like some minor results, minor benefits. Then you follow up 6 months later and you see how the patient is doing. So it would take months to a year potentially to see benefits. Yeah.
So Michael Shanowitz asks, is there a successful example of a therapy requiring editing most of or all target somatic cells?
I'll be interested to see what happens with the eye study. So they were only going after a subset of, I don't know if it was the rods or I forget what cell type it was, but I think they had to hit a lot of that cell type in the eye in order to see a particular, the results they wanted. So yeah.
So how can you prevent, or are you working with big agriculture from monopolizing the technique, making it too expensive for the third world countries?
Yeah, so there's always that concern of big ag, big pharma. Berkeley, as well as MIT and the Broad, have their own licensing agreements with the IP with certain agriculture companies, Bayer or whatever. And there's not as much the scientists themselves can do about that. But when it comes to Africa and small shareholder farmers, they usually have agreements so that the farmers can access the seeds. So it's a case-by-case thing. What we're trying to do at the IGI is address those questions before we start the projects, which is sometimes really hard when you don't quite know where your crop is going to be grown. But you want some agreements up front on, will this be accessible?
How do you ensure unintended or unwanted changes?
Yeah, so we call these off-targets usually. And so that's cutting a sequence of DNA you weren't intending to cut. And this was an issue early on, especially early on in CRISPR research. But over a few years, people were engineering the Cas9 protein to make it more precise. People were using bioinformatics and large screens, large-scale experiments to see what types of guide RNAs minimize off-targets. Where in the genome can we cut so that it doesn't cut elsewhere? So a lot of work has been done to minimize off-targets. Some people also make the argument, well, so when you're filing for a clinical trial, when you're working with the FDA, they want to see that you have 0 off targets, right? They want to make sure you're only cutting what you say you're going to cut. But some people say, you know, every time a cell divides, it introduces mutations. So why should we be better than the cell's normal, you know, division? But people get it down really, really low. And with the sickle cell example, you can actually, when you edit it in outside the lab, you can take a fraction of those cells and sequence them, you know, and make sure you did the edit you want to.
Connie asks, what about Parkinson's disease?
That's a good example. I don't quite know the biology enough to answer that. I don't want to give a wrong expectation. Yeah.
Let's see. Let me direct this to Carrie. There's a question about when will the recording be available and how can I access this? Will this be on the UC Center?
Yeah, we put it out on our newsletter where same place where you got the registration information for this event. We will make that available and make it so that it's not everybody's showing up on the video. You know, you don't have to participate. You didn't get any royalty rights if you were on the video, so we'll keep it secure.
Okay, I've got another question here. Is there a problem of side effects to the Cas molecule if you have to use a lot to get most of a certain type of cell?
Yeah, I guess what I was mentioning before, a body's immune reaction. And we're still learning more about that. And I'm sure as people continue to do clinical trials, things are going to show up. But since we're only now testing on humans, we're going to learn a lot.
Let's see. Masaki Yamoto asks, any comments on using CRISPR to reverse aging? Since we're all retired and old.
Yeah. There's a lot of people with a lot of money in the Bay Area who don't want to age. And so gosh. People are trying. Will any of it work? What they're doing, I don't quite know. But yes, people are trying. There's too much money.
I'll get a good one from Howard Hanayama. How do you know when or how CRISPR is being used for nefarious purposes?
That's a great question. Yeah. So let's take the Chinese twin embryo example. After that happened, people, asked the question, how can we ensure this doesn't happen again? Or if it does, the global community knows. So people have suggested these global observatories that are looking at the international landscape of gene editing. People are also suggesting, and I think there's real momentum, to set up whistleblower pathways. So if you do get wind of something happening, you can report anonymously. So yeah, people recognize that we need mechanisms to both monitor what's happening and be able to report on what's happening. The hard thing, though, is just different countries have different policies. And so what might be not allowed in the US, it might be allowed in another country. So I think what you might see is medical tourism, like we see that now with stem cells. People might go to another country to do a therapy that's not approved in the US. Those types of questions I find interesting.
Let me ask Kevin, are you willing to share the slides? Can you get them also to Carrie?
Sure.
Yeah.
Yes.
Great. We have one other question here. What happened to the Chinese twins? How old are they now?
They went undercover. China was not gonna give their identities, and rightly so. Patient privacy, but I don't think we'll ever know what happened to them. Yeah.
Okay.
Well, let me thank Kevin very much. Ask everybody to clap either on your screen or your reactions. This is
And I-
wonderful.
And I
Okay.
just want to say, we want to thank our president, Bob Kahn. This is his last meeting.
Ah,
And you will have a thank you gift on your porch shortly.
Thanks, Kathy.
thank you very much, Bob.
Welcome.
I hopefully will do half as well as you did in the next year.
And I'll be glad to be working with you as past president. I look forward to that.
Oh, that's great. Maybe you'll be in charge of taking care of our bylaws.
You're already giving out assignments. You can't do that till January.
Thank you, Kevin. It was wonderful. Thank you so
Great.
much.
Thanks for having me.
Thank you so much, Kevin. This is